In 2024, a team of scientists sequenced DNA from a 7,000-year-old mummy, expecting to place it somewhere on the human family tree. Instead, it didn't fit anywhere at all. In 2024, the result appeared in the data with almost no warning. A sequence had been extracted, processed, and compared against the largest genetic reference panels available. The expectation was routine confirmation, a placement within a known lineage, a point on a well-mapped tree. Instead, the result returned empty, no match to any known human population. At first, this was treated as a technical anomaly. Ancient DNA is fragile, often fragmented, and prone to contamination. Errors occur, sequences fail, databases can misalign incomplete data. There are many ways for a result to appear unusual without being meaningful, but this result did not behave like an error. Repeated analysis produced the same outcome. Independent sequencing runs aligned with one another. The genetic markers, though degraded by time, formed a consistent pattern. Each attempt to anchor the sequence within known human variation led to the same absence. There was no clear place for it to belong. Such an outcome should, in principle, be impossible. By the early 21st century, human genetic variation had been mapped across continents and time periods with increasing resolution. Ancient genomes from Europe, Asia, and Africa had been sequenced in growing numbers. Even when individuals belonged to small or isolated populations, their DNA typically showed partial affinities, fragments of similarity that could be traced to neighboring groups or shared ancestors. Human history, as reconstructed through genetics, rarely produces complete isolation. Migration, trade, conflict, and intermarriage leave detectable signals. Even populations separated for thousands of years tend to retain some overlapping markers. A genome without recognizable connection challenges that assumption. So how does that even happen? To understand why, it is necessary to move backward, away from the laboratory and into the landscape where the individual once lived. The remains in question came from a rock shelter known as Takarakori, located in what is now southwestern Libya. Today, the region forms part of the Central Sahara, a vast expanse of arid terrain defined by sand and stone. But the environment at the time of burial was very different. 7,000 years ago, the Sahara was not a desert, it was green. During a period known as the African humid phase, the region supported lakes, grasslands, and seasonal rivers. Animal herds moved across open plains. Human communities settled in areas that are now entirely inhospitable. The boundaries between North Africa and sub-Saharan regions were more permeable, allowing movement across zones that would later become barriers. Takarakori itself was a place of shelter and continuity. Archaeological excavations revealed layers of occupation, including tools, pottery, and evidence of pastoral activity. Among these findings were the naturally mummified remains of several individuals preserved by the dry conditions that followed the region's gradual desiccation. The individuals appeared at first unremarkable within their context. They were identified as part of a pastoral community, likely engaged in herding and small-scale subsistence practices. Their burial practices and material culture aligned with what archaeologists expected from Neolithic populations in the Central Sahara. In this framework, their origins were assumed rather than questioned. Existing models of North African prehistory suggested that populations in the region were shaped by multiple waves of migration. Some groups were thought to have moved northward from sub-Saharan Africa during humid periods. Others may have arrived from the Eastern Mediterranean or the Near East, bringing new technologies and subsistence strategies. Over time, these movements were believed to have produced a network of interconnected populations, each contributing to the genetic landscape of the region. Before the introduction of genetic analysis, such interpretations relied on three primary forms of evidence. One was material culture, tools, pottery styles, and settlement patterns that could be compared across sites. Similarities in these artifacts often suggested contact or shared origins, though such connections could also arise through imitation or parallel development. Another was environmental reconstruction. By studying ancient climate patterns, researchers could infer when migration corridors were open or closed. Periods of increased rainfall would have facilitated movement across the Sahara, while arid phases would have restricted it. The third was physical anthropology. Skeletal remains were examined for morphological traits such as cranial shape and dental patterns, which were used to classify individuals within broader human groups. These methods, while informative, were limited by subjective interpretation and the overlapping nature of human variation. Within this framework, the individuals at Takarakori were placed into an expected narrative. They were part of a known population, participating in known processes of migration and cultural exchange. There was no reason to suspect otherwise. Until the DNA. The introduction of DNA analysis offered a new approach. Rather than inferring relationships from artifacts or physical traits, researchers could examine genetic material directly, comparing ancient individuals to both modern populations and other ancient genomes. This method had already transformed the study of human history. Ancient DNA from European sites had revealed complex patterns of migration during the Neolithic period. Studies of early farmers and hunter-gatherers showed that cultural transitions were often accompanied by significant genetic turnover. In Egypt, genomic analysis of mummies had identified affinities with populations from the Eastern Mediterranean, aligning with historical accounts of regional interaction. In most cases, DNA analysis did not overturn existing narratives so much as refine them. The expectation for the Takarakori remains was similar. Researchers anticipated that the genomes would cluster with known North African lineages, perhaps showing connections to sub-Saharan groups or to populations from the Nile Valley and beyond. The process began with careful extraction. Ancient DNA is often preserved in dense bones or teeth, where it is protected from environmental degradation. Samples were taken under controlled conditions, with strict protocols to minimize contamination from modern human DNA. The extracted material was then sequenced, producing fragments that could be assembled into a partial genome. These sequences were compared against reference databases, containing genetic information from a wide range of populations. It was at this stage that the first anomaly appeared. The sequences did not align with expected clusters. They did not show clear affinity with sub-Saharan African populations, nor did they match known North African or Near Eastern lineages. The statistical models used to estimate ancestry produced inconsistent results, failing to assign the genome to any established group. At first, this was treated with caution. Ancient DNA studies often involve uncertainty. Degradation can obscure signals. Missing data can distort comparisons. It is common for early analyses to produce ambiguous results that are later clarified with additional data. So the process was repeated and repeated again. New samples were taken. Independent laboratories conducted parallel analyses. Different computational models were applied, each designed to detect patterns of relatedness within large datasets. The outcome remained the same. There was no meaningful match. As the data accumulated, the interpretation shifted from uncertainty to a more defined problem. The genome did not simply fail to align neatly within existing categories. It appeared to represent a lineage that diverged from other known human populations at a much earlier point. Estimates suggested a separation dating back tens of thousands of years. This raised a series of questions that extended beyond the individual. If this lineage had diverged so early, why had it not been detected before? If it had persisted in North Africa for such a long period, what had happened to it? And how could a population remain genetically distinct in a region characterized by movement and exchange? Initial explanations focused on technical factors. Contamination was considered, though the consistency of the results across multiple tests argued against it. Modern DNA tends to introduce recognizable patterns, often aligning with contemporary populations. The sequences in question did not resemble modern contamination. Degradation was another possibility. Ancient DNA can fragment in ways that obscure true relationships. However, the quality of the data, while incomplete, was sufficient to identify broad patterns of ancestry. The absence of a match could not be explained solely by missing information. Attention then turned to the limitations of reference databases. Genetic comparisons depend on the availability of data. If a particular lineage is not represented in the reference set, it may appear as an outlier. In this sense, no match does not necessarily imply uniqueness, but rather absence from the known sample. Yet the scale of existing datasets complicates this explanation. Modern and ancient genomes from across Africa, Europe, and Asia have been extensively cataloged. While gaps remain, the probability of an entirely unrepresented lineage of this age is low, though not impossible. Another possibility is that the lineage once existed, more widely, but has since disappeared. In this scenario, the Takarakori individuals would represent a remnant of a population that was later absorbed or replaced by other groups. Genetic signals from such a population might survive only in diluted form, too faint to be recognized as a distinct lineage in modern data. There are precedents for this kind of pattern. Studies of ancient populations in other regions have identified ghost lineages, inferred from genetic data, but not directly observed in the archaeological record. These lineages are reconstructed through their influence on other populations, even when no direct samples are available. In the case of Takarakori, however, the lineage appears directly, yet without clear descendants. This creates a tension between the physical evidence and the genetic data. The individuals themselves lived within a known cultural context. Their tools, burial practices, and environmental setting align with established models of Neolithic life in the Sahara. There is no indication that they belonged to an entirely separate or unknown civilization, and yet their genetic profile suggests a deeper separation. This tension has led to differing interpretations among researchers. Some emphasize the continuity of human behavior, arguing that cultural similarities indicate interaction and shared practices regardless of genetic distinctiveness. In this view, the Takarakori individuals participated in the same networks as neighboring groups, even if their ancestry was different. Others focus on the genetic evidence, suggesting that the Sahara may have hosted a greater diversity of populations than previously recognized. The humid period could have allowed multiple groups to coexist, each maintaining a degree of isolation despite geographic proximity. A more cautious position acknowledges both perspectives while emphasizing the limits of current data. Genetic analysis, while powerful, is not definitive. It provides probabilities, not certainties. The interpretation of ancient genomes depends on models that are themselves subject to revision. As new data becomes available, patterns that once appeared anomalous may find context. At the same time, the absence of a match remains significant. It suggests that the human past contains structures that are not yet fully mapped. The familiar image of a branching tree with clear lines of descent may be an oversimplification. Instead, the history of human populations may resemble a network with nodes that appear and disappear, some leaving clear traces, others fading almost entirely. In this sense, the Takarakori genome does not resolve a question so much as reframe it. Rather than asking how a known population fits into existing models, it raises the possibility that the models themselves are incomplete. The categories used to describe human variation, regions, cultures, lineages, may not capture the full complexity of past populations. This does not imply that the individuals were fundamentally different in a biological sense. They were human, sharing the same broad characteristics as other Homo sapiens. The differences lie in the patterns of ancestry, in the pathways through which genetic material has been transmitted across generations. Those pathways are shaped by events that are often invisible in the archaeological record. Small populations can persist in relative isolation for long periods, especially in environments that limit contact. When conditions change, through climate shifts, migration, or cultural transformation, such populations may be absorbed or displaced, leaving behind only fragmentary evidence. The Sahara itself provides a context for such processes. As the humid period ended, the region gradually returned to aridity. Lakes dried, vegetation receded, and human populations moved toward more stable environments. In this transition, communities that had once thrived in the interior may have fragmented, merged with others, or disappeared entirely. The genetic legacy of these processes is uneven. Some lineages expand and become widely represented, others contract and vanish. The survival of a lineage in the genetic record depends on a combination of chance, environment, and historical circumstance. The Takarakori individuals appear to belong to one of the latter cases. Their DNA captures a lineage that persisted for tens of thousands of years and then, for reasons that remain unclear, did not continue in a recognizable form. This does not close the story. If anything, it suggests that similar cases may exist elsewhere, waiting to be identified as techniques improve and datasets expand. Each new genome has the potential to alter the structure of the human past, not by providing definitive answers, but by revealing new questions. The initial result, no match, remains. And that might be the most important part. It no longer reads simply as an anomaly, but as a gap in our understanding, a missing piece of the human story, a lineage that existed and then disappeared. And the only reason we know it ever existed at all is because, for a brief moment, it showed up where it wasn't supposed to.